Hello student, today we talk about the taxonomic procedure. Taxonomic procedure, it's kind of preservation of animal for their identification and classification, because for just getting the information is not useful for the identification of such animals. We have to see that animal as in pictures or in specific preservation stage, because these animals identification more practically is important for the identification, because practical knowledge is more than the theory knowledge. So we have to identify the animals by knowing their body structure, their colors, their living habitat, feeding habitat, their geographical area where they found. So all this information we have to collect, so that for better understanding the species concept, so that we have to proceed them in to preserve them species for the such a identification and classification of them. So in today's topic we talk about the how animals are collected for the preservation and to prepare in the museum for the identification and better study for the human being. So basically, in today's topic, we're talking about the how taxonomic procedure, that is first part, that is collection, how these species are collected for the preservation and their identification. So let's start today's topic, that is taxonomic procedure part first. So basically, taxonomic collection is a biological collection which is typically preserve the plants or animal specimen along with the specimen documentation such as labels and notation. So basically, taxonomic collection is a type of biological collection. So in biological collection, we preserve the plants and like like fishes, insects, and different kind of species of birds, animals, and this species documentation along with their documentation where this species is found, how its living habitat, feeding habitat, its labels, where the date of collection, preservation. This information is also with this preserved plant and animal is found. So these preserved animal and plants are kept in a museum for their identification, and the knowing the species on the earth is better for the human being for their identification and preservation. So in today's topic we're talking about the methods of collection, how the animals or plants are collected from the different geographical area for their preservation and their identification. First point is a mist net. Mist net is useful for the collection of aerial birds, because these birds are found in a aerial spaces, and they are and humans are unable to catch such aerial birds, so that the scientist or the taxonomic collector use the mist net for catching the migratory birds. So these migratory birds move from one place to another place, and these migratory birds or different kind of birds useful for their knowing the structure, function, their physiology, their anatomy, and their body structure, morphology for the human study. So we have to collect such a birds for the human study. So mist nest is useful for the collection of birds. Next part, that is UV light. So UV light is useful for the attracting the nocturnal insects. So for the collection of insect, this UV light is useful. Mostly many insect can see the ultraviolet light, which has the shorter wavelength than the light visible to the human eye. We know that the UV lights cannot visible for the human eyes, but insects see this ultraviolet light which having this short wavelength, because human see the long wavelength light, but short wavelength UV light containing the very short wavelength, so that human eyes cannot detect the UV light. But these nocturnal insect or insects seeing this ultraviolet light and attract towards this light. So for this reason, a black light will attract the different insect than the regular incandescent light. So we know that regularly insect attract towards the light, but normally the UV light is more attractant for the insect, so that the taxonomic collector use the UV light for the attraction of the insect. Basically, the black light can be suspended in front of white sheet and giving the flying insect a surface on which to land. So basically, the light is suspended in the front of white sheet so that the insect can be sit on that such surface, so you can then absorb the insect on sheet and collect any interesting specimen by hand. So the black light trap is constructed by suspending a black light over a bucket or other container, so usually this funnel with the funnel inside. So basically this UV light is suspended in the on the white sheet with the collector. So this collector collecting the insects which based on the land surface of the white sheet. So basically this constructed for the collection of the nocturnal insect. So insect fly to the light, fall down through the funnel into the bucket and then trap inside the container. So black light trap they sometimes contain the killing agent but can also be used without one to collect live specimen. So we can use these nocturnal use this UV light attractant for the collection of insect by using the killing agent or getting the live specimen of the insect, because different kind of insect are studied for the in biology. In zoology we study the different kinds of insect in entomology. So these insects are important for the study of entomology and they are collected by the using the UV light attractant. Next part is that is Melis traps. So Melis trap is a large tent-like structure and mostly this structure is used for the trapping the flies and wasps. So basically butterflies, wasps, these are insects which are collected in the Melis traps because they didn't attract towards the UV light. So for the collection of such a flies and wasps, malice traps are usually used by the taxonomics. So insect fly into the tent wall and funnel into the collecting vessel attached to the highest point. So in this figure you can see that this is the nest. So all these are the nest and this is the collecting point. So this is the collecting vessel where the all the insect are collected in the vessel. So malice trap is mostly useful for the trapping the flies and wasps and collection of them. Also insect are collecting by the beating and sweeping. So we know that the insect are live on the plants, their leaves, their stems or on the grasses. So by you on daytime different kinds of insects are also found, so they didn't attract towards the UV light. For that we have to collect such an insect by beating and sweeping. So beating and sweeping is kind of collecting technique for the diurnal insects. Next part that is aquatic insect, how aquatic insect get trapped. So this is the plankton net and this is the deep net. So these two nets are usually used for the collection of aquatic insect and other arthropod by getting collected by the using deep nets and plankton nets. Mostly these nets are useful for the collection of aquatic insects and the arthropods. Next that is trawling and dredging for the collecting deep sea animals. So this is trawling and this is degrading, which useful for the collection the deep sea animals. So in this figure you can see that this is the net which collect the organism of the deep sea. This is also the useful for the collection of deep sea animals, and this collection of animals mostly didn't hurt that animal or didn't damage the body parts of animal, because we have to collect such animals in a entire body structure or entire morphology without damaging them, so that we can identify or classify and study them betterly. Next part that is collecting net. Collecting net is also essential or usually used for the collection of insect on the aerial insect which are flying over the air. So these collecting net are useful for the collection of insect and wasps. Next part, that is aspirator. Aspirator is a type of tube. These tube are mostly useful for the collection of insects. So basically nocturnal insects are collected by this aspirator tube. This aspirator tube having one metal tube and one plastic tube. So this plastic tube is useful for the inhalation, and metal tube is useful for the collection of insects from the surface of the white sheet which containing the UV light. Next part, that is flotation method. Flotation method usually used to collect the arthropods, eggs, pupae of insect from the soil and matted vegetation. Basically these eggs and pupae are live in the soil or the matted vegetation. So we have to collect such a soil which containing such egg and pupae and dip in a water, so that these eggs and pupae can float on the surface, and then we easily collect such a eggs and pupae. That is flotation method. So flotation method means the soil is deep into the water, then these eggs and pupae for by their light weight they get upward, and then we easily collect them. So that is flotation method. Then next part, that is killing bottles. So killing bottles containing the poison. So this poison kill the insects or beetles. So for the collection of insects and beetles usually used for the killing purpose. So mostly these tubes or bottles are used for the collection of insect during the UV light traps. So that is for today. So in today's lecture we see the different collection methods of animals. So these different collection methods such as a mist nest, aspirator, flotation method. So these methods are useful for the collection of animals, insects and different kinds of species. So this collection of animal is useful for their identification and their preservation. So that is for today. Thank you.